this way. All right, and then can I move this one? Let's move this one over here so it's out of my way. All right. Okay. All right, so in the Griffith experiment, we should understand a couple of things. All right. Okay, so here we go. All right, so what were we studying in the Griffith experiment? What were we studying in the Griffith experiment? What was the relationship that was being studied? Okay, so thank you, Sam. Sam said bacteria. Good job. Okay, we were studying bacteria. Excellent job. All right, so we're studying bacteria. All right, so there it is, bacteria. All right, now that's what we're studying. Okay, now does anybody know? Most of you guys think of bacteria and you hear the word germs. So that's what this guy was studying. He was studying germs. Now, Keith, how many types of germs was he studying? How many types of bacteria?
He was studying 2 bacteria.
Exactly, right? So what were the 2 types of bacteria you're studying, Anthony Gonzalez?
I'm not sure.
Come on, bro. You need to be sure. Just say, I don't know, Rodriguez. I didn't do any of my work. Okay, so, okay, Ariel, go ahead and tell Anthony G, AG, what were the 2 types of germs? What are the 2 types of bacteria that you're studying?
The 2 types of bacteria were the harmless bacteria and the disease bacteria.
Good. So what did he call, what did the harmless bacteria look like?
It had rough edges or rough colonies.
No, I don't think so. It was the opposite. I'm pretty sure. So let's double check, because that's why I think my video's messed up. So if I go to the actual text here, okay, just that's why I want to make sure we do this live here. So that way you guys, there's no confusion, right? So here's the Griffith experiment. If you ever get lost, here's the text, okay? All right, so here we go, look. Disease-causing bacteria are the smooth colonies. Make sense? Yes, yes, yes. So let me go ahead and put that in there. So these guys, I put it in here. All right, so let's pretend that this purple marble is the smooth bacteria. All right, it's the smooth one. They look smooth. All right, what's the other one? And
The rough bacteria.
The rough bacteria, right? So if we put it in, here we go. Let's take a little picture here, right? So here's the other one, the rough, harmless bacteria. All right, whoops, it went the wrong way, I bet you, dang it. Okay, so here we go. Control C, let's put it back over here, boom. All right, so there it is, right? Okay, now, here's what you need to do. There's 2 types of germs, just like Keith said. Very good. All right. So there's 2 types of germs. So 2 types. All right. There's 2 types. Okay. The smooth and the rough. Okay. Now you guys need to know, what do these germs do to the mice? What is this purple? What do the smooth bacteria do to the mice?
It causes the mice to develop pneumonia.
Exactly. It causes them to develop pneumonia. Very good. And then therefore they what?
Die.
They die. Exactly. Right. So let me go ahead and put a little picture. So what ends up happening is they get injected in, and these dudes, these poor little mice, die. Right? So I don't know if you can see that, but they die. All right, they die. These guys, when they get injected, what happens to them?
They live.
They live, exactly, right? So let me go ahead and take a picture of that. So you guys have it. So these guys, okay, when you inject the other guys, the mice live. All right, so here we go. Here we go. Okay, there it is. All right, does it make sense? Are everybody with me on this? All right, okay, all right, cool. All right, so now All right, so that's what this guy did. He wanted to figure out what's the deal with these germs, right? And therefore figure out he was studying pneumonia and these germs, right? But what he ended up doing is he ended up doing something in his experiment that he didn't expect. Okay, so what's the next step that he did? What's the next step that he did? What's the next step? What did he do next?
He heated the disease bacteria.
Yeah, so think about it like this, all right? So I'm gonna show you guys, okay, some visuals here, all right? So check it out. So if I go back to my camera here, right? Imagine that he had like this little flask with water, right? And what he did is he took these guys, right? And he added them into the flask, right? So if I'm looking, there they are, right? Okay, they're chilling in the bottom. All right, so now what he ended up doing is he heated this up. He heated this up, okay? Why would he want to heat that up? Why would he want to heat this up?
To burn all the disease bacteria.
Right, you're right. Well, think about it. Okay, Keith, have you ever do you like chicken?
Yes.
Do you eat raw chicken?
No.
Why not?
Because it's bad for you.
Because what?
It's bad for you.
It's yeah, why though?
Because it's non-cooked, which means if it's not cooked, it's raw. When it's raw, it's just bad. You get a bad taste.
what is-
and
What is
You
it?
can
What
get
is it
sick.
when it's raw? What does it have on it? What does it have on it when it's raw?
oh, it has like miniature. When it's raw, it has like little parasites all over the. You have,
Dude, it's on the screen. When okay, when what does raw chicken have on it? Josiah, tell him.
Disease causing bacteria.
Yes, germs, y'all, germs. So when you cook the chicken, yes, the chicken. Okay, when you cook the chicken, okay, you kill the germs. You see what I'm telling you, right? And so what ended up happening is this scientist, Dr. Griffith, right? What he did is he took some heat, right? Let me get the heat. There it is, right? Control C. What he did was he took some heat, right? And he heated these bad boys up. He took the smooth bacteria, he heated it up, and guess what he did after that? Guess what he did?
He injected it into the mice.
Yes, he injected it into the mice. Now, what do you think happened to the mice? Did the mice live or did the mice die?
They lived.
They lived. That's right. They lived. So when he injected this juice that he heated it up, the mice, oh, it's my bad. There we go. I'm making mistakes again. Okay. What ended up happening is, well, let me get this. There you go. Let me actually delete it. Right. So take this right here. Okay. The mouse lived. Does this make sense? Yes or no? Are you guys with me on this? Are you guys with me? Right? Okay, so let me take a picture of this, blow this thing up. All right, let's blow this up. And let's put it on a new one. Here we go. This is where we're at. All right, are you guys with me? Are you with me, Sam? You with me? All right, so Anthony, do you with me? Are you with me, Anthony?
Yeah,
How many types of germs did we have? Did he study?
2.
Good. What did he do with what did he do with the smooth bacteria in the flask?
He heated it up.
He heated it up and then he took the juice, injected that into the mice, and what happened to the mice?
They survived.
They survived, right? So now, guess what he did now? Guess what the next step was, Keith? Guess what the next step was, Keith, Matthew? What do you guys think?
He injected the other one into the mice again.
No, he did this. He took this, right? Now that this is heated, right? He heated it up, right? And now he added these guys. What are these? What do the yellow marbles represent?
The rough bacteria. That's harmless.
Yeah, the harmless. So he added the harmless in there. Oh shoot. Busting up my electronics for y'all. Okay. So he added them in there now. Look. Okay, and then let me shake it out a little bit. Hold on. All right, anyways. Okay, so now he took this now, right? He took this. Let me take a screenshot of this. Let me take a screenshot. All right, here we go. Here we go. Take a screenshot of this. So you guys have it. Bang. There it is. Copy. Right. All right. And now I can put it down because it's heavy. All right. And there he is. Look. So now he has the mixture. Okay. He has the mixture. All right. So he got the dead bacteria. Right. So this is the dead bacteria here. The dead, the heated, these guys that are kind of dead, right? And then he added the live. So what should happen to the mice now? What do you guys think? What do you guys think?
It died.
Okay, what should happen to the mice?
Does it die?
Hold up. You think they should die?
Yeah.
Why would they die look, so the yellow ones don't kill the mice. The juice that got heated doesn't kill the mice. So when you add the two together,
Oh, I thought you put the purple in it. Oh, my fault.
no, the purple's already is the heated juice,
Oh, okay. Sorry.
right? The purple is the heated juice. So what should happen, y'all?
It stays alive.
Huh?
It stays alive.
The mice should stay alive technically, right?
Yeah.
Right? But guess what? The mice actually did what?
Died.
They died. They died. So when he added the heated killed bacteria, so he added this. So when he mixed the two together, right? So when he took this mixture, so he took, let me see here. He took the mixture. So this right here, that doesn't look good. Okay, how about yellow, red? So when he took these two guys right here, this guy and this guy, and he added them together to give you this stuff got injected, the mouse died. What the heck? Why did the mouse die? Why did the mouse die, y'all? Crazy. Think about that. Why did the mice die? Why did the mice die? See if I can get my Zoom to work here in my class so I can show you my whiteboard. Sign in. Google. Continue. What do you think, Ariel?
I think some genes weren't killed while being heated. Some are left just a little bit, and then once they got mixed with the harmless ones, they just started to what's it called? They started to grow more.
There you go. I think you're on the right track. See here. Okay, here we go. I'm zooming in. See, can I join my own personal meeting? No, I can't. Meetings. Start. Am I joining? Let me see if I'm joining. Oh, I'm in. Okay. Whoa. Okay. All right. Good. All right. So share content. Can I share content? Photo screen? Nope. How about, can I share the screen? Okay, let me see. Am I sharing the screen? Are you guys seeing me? Trying to mess around here. Okay, hold on. You are sharing your screen. So where is it? So if I go like this. Can you guys see? No, you guys can't. Can you guys see what I'm looking at on my phone?
No.
No.
No? All right, never mind. That's weak sauce. Okay. All right, so here's the deal, right? So let's go back to this. All right, so here's what we got. Okay, so we got these two. What you need to understand about a cell is basically the following, okay? A cell is just basically a collection of molecules, all right? So when we're looking at the R strain, right? The reason why the R strain kills doesn't do anything to the mice is because it's making, well, first of all, the L's, okay, the L's, guys, okay, those are lipids, okay, they're lipids. The G's, that represents the glucose, like cells need glucose for energy, right? The P's represent proteins, okay, proteins. And think of proteins like apps, okay? And then you got the DNA, okay? So every single cell has DNA, right? So what ends up happening is, remember, DNA has codes, and the codes allow the cell to make the protein. And so basically what the R strain does, it doesn't have any proteins that are going to cause the mice to die. So that's why the mice live. On the other hand, the S strain is going to produce proteins, and these proteins are going to cause the mouse to die, right? Okay, it's kind of like the same reason why some of you guys have, like, I have light skin, because my DNA uses has codes to make proteins that gives me light skin. Okay, Josiah has different DNA that's giving making him build a different protein that's giving him a different type of skin, right? And so that's why these bacteria act the way they do. And so what you guys have to imagine is that when this bacteria got heated, when the S strain got heated, look what happened to the cell. What happened to the cells? What happened?
They spread it apart.
Yeah, the cell basically broke. Like, it literally disintegrated. So the heat disintegrated the cell. So instead of having the lipids building the cell membrane, okay, they scatter. There's no longer a cell membrane. All the molecules are just basically floating in the juice, right? So you have to imagine that all the molecules are in the juice. So what ends up happening is when he added these guys now, okay, so when he adds these guys now, okay, let me see here, let me erase all this. So now when he takes this guy right here, remember, the rough doesn't do anything to the mouse, right? But when he took, okay, the S strain, he heated it, basically it disintegrated those cells. But then what ends up happening is when he adds this guy into the juice, okay, now guess what happened? What do you think happened? What do you think happened? So this is the R strain right here. Okay, what do you guys think happened? Take a guess.
Is it separated too?
No, no, because remember the heat's done. Now the heat's done. So that's
Oh.
so the heat. So now there's no more heat. Good, no more heat, right? We took it off the heat. Okay, the S strain, the bad bacteria disintegrated. They're just like all the pieces are just all like, right? And then we added the R strain, right? So no more heat. Okay, good. Actually that's a good question right there. So no heat. We remove the heat, right? What happens next? Because remember what happened when we took this mixture and we injected the mice. What happened to the mice?
It lived.
No,
It died.
the mice died. When we added this, the mice died. So why did the mice die? That's my question. That's my question.
Wait, what was the question again? One more time.
Why did the mice die? Because think about it. Look, we
Oh,
have the killed R strain, right? We have the no, I'm sorry. We have the killed the killed smooth strain, right? So these bacteria over here killed the mice, and then we have the bacteria mixture, like we mix in the ones that don't do anything as well. So technically both of these things by themselves don't do anything to the mice. But when we actually did the experiment, the mice died. Why? Why? Let's blow this up a little bit. Let's blow this up. Okay, so you guys can see it because I know it's hard to see on the Zoom. What do you guys think happened? What do you think? Whose DNA is this? Whose DNA is that? The harmless or the harmful?
Harmful.
The harmful, very good. This right here is the S strain DNA. This guy has the codes to make the proteins that cause pneumonia. So what happened was somehow that DNA got into the harmless bacteria. And what ended up happening is now this harmless bacteria somehow got transformed into S strain bacteria. And that's why this mixture killed the mice, because somehow this DNA got into the harmless bacteria, and that DNA from the S strain transformed this guy into a harmful bacteria. Okay. So let me draw it to you guys another way. Okay. Let me draw it another way. So here's the smooth strain. Okay. He's harmful, right? And let's put some DNA in him. Right, there he is, right? When you add that to the mice die. I should do it on a piece of paper. It's probably easier. Okay, actually, you know what? Yes, I'm gonna do that on a piece of paper. Hold on. This is annoying me to no extent. Okay, so here we go. Let's do it on a piece of paper. All right, check it out. Let's see if this works. Apologize for the technical difficulties. Okay, here, oh, there's a paper. We're gonna go old school. We're gonna go old school, Keith. We're going old school. Okay. All right, here we go. Let's move the mic a little closer to me. Hopefully this makes sense. All right, okay. You still hear me? You guys still hear me? Yes? Okay, all right. So here's what's happening, all right? So check it out. So remember, there's two types of germs. All right, so that's what we have. There's two types of germs. Okay, so here we go. Zoom in a little bit. Okay, so you got the smooth strain. Okay, let's just say it looks like this. Right? And this guy has DNA that makes them like harmful. And so when this guy goes into the mice died. Okay, super important. Then there's this other bacteria. Okay, they call them the rough. And so he looks like this. Okay, and I'm gonna put him as a happy face, right? This guy has different type of DNA. Okay, and that DNA makes him harmless. So when he goes into the mice live. Super important. But now what the scientist did is he took a beaker, right? He added these guys in there, added some heat. And what happened to these bacteria right here? What happened? What happened to these guys? What did the heat do to these guys?
It killed them.
It broke it.
They got broke or they died, exactly. So I'll put X's. They're dead. And then he injected that into the mice. The mice lived. Right? So then here's what he decided to do on the fourth trial. He decided to take this beaker that had these dead guys in there, and he added the live harmless bacteria. Does that make sense? And then he injected the mice. The mice did what, Josiah?
They died.
They died, exactly. But here, somebody explain to me why that doesn't make sense? Explain it to me. Ariel, explain it to me. Why doesn't this make sense?
It doesn't make sense because both the heated bacteria and the harmless bacteria caused the mice to live, but when mixed together, the mice died.
Exactly, it doesn't make sense, right? When you add two things, right? When you add this guy and this guy, right? When you added them, right? The mice should live. But instead, the mice, they died. Does that make sense? And Keith, I just told you why the mice died. Why did the mice die? What must have happened? Remember, here's the beaker. Here's the rough guy right here. And remember that dead bacteria, okay, he's basically dead and he broke apart and he has his DNA in here. So somehow this DNA got into this guy. And now, and so what ended up happening is when this stuff went into the mouse, okay, the mouse died. And so what, guess what the doctor did, okay, or the scientist? He opened up the mouse. So here's the mouse. I don't know if I can open up, I don't know if I can draw a mouse. That looks like a cat. But he looked at his lungs. Right? And he took a sample of his lungs and he opened them up and when he looked at his lungs, guess what he found under the microscope? Guess what he found in the mouse's lungs? Did he find these guys or these guys? What do you guys think?
Bacteria.
He-
The
he-
disease bacteria.
yeah, he found in the lungs these guys. He found these guys in the lungs of the mouse. When he examined the mouse, okay, Griffith examined the mouse and he found these guys. Where did these guys come from? If they were dead, remember, these guys were dead in the juice. What happened? What happened? Where did they come from? That's super important.
They came from the rough strain.
Say that again. Say that
They
again.
came from the rough bacteria.
Okay, say it a little bit. Samuel, what happened? What happened? Where did these guys come from? If they were dead in the juice, how the heck were they present in the mouse that was dead? Exactly. So remember, when this DNA, somehow Griffith realized that there must have been something that got transferred to the harmless bacteria. And then, okay, this DNA that got transferred transformed these harmless bacteria into harmful bacteria. And now that's why the mice died, because the DNA transformed. Think of it it's like, I don't know. Yeah, it's like what? Like sort of, I think of it more like, it's more like this, or like, okay, like we're talking about cell phones for the longest, right? Okay, cell phones, right? If I have two phones, right? And if I have two phones, and this one I have Twitter on this, but this one I don't have Twitter. What do I have to do to the one that doesn't have Twitter on it? What do I have to do? Download what?
You have to download Twitter.
Exactly. So basically, that's exactly what happened here. Okay. Basically what this harmless bacteria did is, when he got mixed in the juice, he basically downloaded the DNA from the harmful bacteria, which transformed them into harmful bacteria. Does that make sense? Okay, that's why I talk about DNA. The DNA right here, those are codes. And when those codes get downloaded, it transforms you and gives you new abilities. Does that make sense? Ariel, are you with me on this? Yes, 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 yes. Okay, so explain it to me. Explain it to me. Someone explain it to me. So step one, what happened? How many types of bacteria did we have?
2.
2, good. Which one killed the mice?
The smooth bacteria.
Good, okay. Now, what did he do to the smooth bacteria right here?
He heated it.
He heated it, and then what? And then this juice, what did it do to the mice?
Let it live.
They lived. Very good. Then he took this juice with the dead bacteria, right? That's why we cook our food, to kill bacteria. He did the same thing. Now he added the live, harmless bacteria. And what happened to this harmless bacteria when he was in the juice?
It copied something from the old bacteria.
Not copied. Think of it like this.
Downloaded
It downloaded what?
something from the
Not something.
DNA
The DNA from the,
from the smooth bacteria.
exactly, from the dead bacteria, that the harmful. And when it downloaded it, it transformed this harmless bacteria into harmful bacteria. And so that way, when it got injected, that's why the mice died. Because somehow, because not somehow, the harmless bacteria literally got transformed. That's why this experiment is so famous, because that's the first time people realized that there's molecules that can transform, that have codes that can be downloaded, and they can transform living things to do other things. Like, guys, you guys live in an era where your kids, one day, you guys are going to be able to see like genetic engineering at the human level. Like, we're talking about like Anthony, let's say he wants to have a son, and he's like, you know what? I want my son to have blonde hair. And there might be a time where maybe not Anthony as a dad, but maybe Anthony as a grandfather, like where his grandkids be like, hey, dad, guess what? There's this thing where you can go to the doctor now, and you can ask the doctor to put a gene into your sperm cell so that you can have kids that have the traits that you want. Like, it's crazy. Like, we are literally getting to that point where we're going to be like, we do it already with things that are not human beings, okay, like plants and animals, but we have this technology to download codes into cells and transform those cells into something different. And so what we're going to do this semester is really try to understand, okay, how we're going to, how does DNA hold codes? Write that down. How does DNA store codes? That's what we're going to get into. All right. Give me a thumbs up if this helped, hopefully, and made sense towards the end. Yes. Okay, hopefully the drawings helped on the paper. I didn't realize I should have like done it on the paper. Like, I haven't done that in paper in a long time, but hopefully that works. Okay. Any other questions, concerns, guys? Any other questions? All right, if you guys don't have any other questions, I'm gonna download this video into YouTube, and then I'll have it for you guys to review. And then we'll talk about it more about it tomorrow. For homework. Okay, for homework, hold on. Let me see here. Hold on. Mono share, live, disable, stop